we are presenting the research project named siRNAs targeting NRP-1 as a new therapeutic target for COVID-19, which was developed at the Medical School Instituto Politécnico Nacional. To begin with, let's talk about COVID-19, which is a disease caused by the SARS-CoV-2 virus and has become an international health emergency in the world, with 767.4 million accumulated cases and 6.9 million deaths, with the Americas being the most affected region. The main symptoms of COVID-19 include cough, tiredness, nasal congestion, fever, headache, sore throat, and shortness of breath. And the main risk factors are hypertension, diabetes, obesity, or the gestational period. But the main question here is, how does the SARS-CoV-2 virus enter the cells? This is due to the binding of the S protein of the coronavirus variant to the angiotensin II convector enzyme and subsequently to the neuropilin-1 receptor, forming the key log structure complex that allows the virus to enter the cell and being viral replication. Once the virus enters the alveoli, it produces a lesion in the alveolar wall which generates an inflammatory reaction and leads to the activation of general immunity through the activation of macrophages by means of cytokines, phagocytosis, and cytotoxicity, which is turned generate alveolitis, produce edema, and generate pneumonitis, as well as alterations in the same vessels, which generate small thrombi and alter blood perfusion. There is scientific evidence that melittin, which is a V venom peptide, binds to the neuropilin-1 receptor in order to enter the cell, which is why we designed an animal model with melittin to simulate the entry of SARS-CoV-2 in the cell, given that they have in common the binding to NRP-1 in order to enter the cell, and both produce alveolar inflammation. Therefore, the aim of the project was precisely developing and evaluating siRNAs targeting neuropilin-1 as a potential therapeutic option for COVID-19. Once the siRNA is synthesized, it is introduced into the cytoplasm in an endogenic manner, and this molecule at the same time incorporates the risk-silencing inducer complex, which allows the separation of the siRNA strands to achieve, in this case, a specific gene silencing. To develop this project, we used bioinformatics tools for the design of the siRNAs, after which the synthesized of the sequences was carried out in the Maydel equipment and the evaluation by PCR in real time. Finally, the melittin model was developed for the evaluation of the designed siRNAs. As results, we present the design and synthesis of the siRNAs targeting NRP-1. For this, We searched for the coding regions of each variant in both species. We obtained the secondary structure of each of the variants in both species. The sequences of the designed siRNAs were obtained, along with the psychomedical parameters including melting temperature, total concentration, and the percentage of guanine and cytosine. With the designed sequences and the previously predicted secondary structure, we located the hybridization sites of each siRNA. We observed that the subsequently hybridization sites are located in loops and stem loops, providing successfully specific gene silencing. The expression of the siRNAs targeted to NRP-1 was determined 72 hours after their administration, and it can be observed that the expression of NRP-1 decreased significantly in the group administered with siRNA compared to the other experimental groups. Similarly, the physiological parameters were determined after the administration of the siRNAs at both time 0 and 3 days post-administration. It was found that there were no significant changes in the physiological parameters due to the administration of the siRNAs. In the Maydel model, the effect of siRNA administration was evaluated by measuring lung lesions using image analysis software ImageJ. It was observed that in the Maydel model without siRNA administration, there was a higher number of lung lesions compared to the group treated with siRNA. This indicates that the administration of siRNA has a protective effect and reduces the extent of lung damage caused by Maydel. If we observe the Maydel model with siRNA administration, we can notice that the siRNAs have a protective effect and decrease the percentage of severity of the affected area. This indicates that the administration of siRNAs can attenuate the damage caused by the Maydel and reduce the extent of lung injury. Graphically, the percentage of lesions without siRNA administration and with siRNA administration can be observed. It can be seen that the percentage of severity in the affected lung area decreases when the siRNAs are administered. Based on the findings, it can be concluded that the siRNA targeting the NRP-1 receptor effectively decreases NRP-1 expression, indicated its potential as a therapeutic option for COVID-19. The designed and synthesized siRNAs demonstrate a significant protective effect on lung lesions, suggesting their potential application in mitigating the severity of the disease. Finally, we gratefully acknowledge to Instituto Politécnico Nacional for the research grants Proyectos Insignia IPN 2015 SAP 202311313 and CONACYT CVU 8902 3.7 for the support of the scholarship granted.